introduction and for the possibility to speak here and give this, this lecture. I hope that in an hour or so, or less than an hour, this title will have become clear. I want to show you some sort of basic statistics and how it is used and reflected in biological applications, but not only biological. The reason why I picked this is because it's really quite important in a whole number of fields because the thinking is absolutely generic. Much of the work, I'm not going to speak much about our own research, a little bit, but the whole topic was shaped in our group in discussions with the students, Juliane Perner and Marc Saganbari, and the postdoc, Julia Lasserre. And several of the slides actually are stolen from them, so I owe them a lot for this. For those of you who are not really into biology, a quick rehash. I think this is now more or less common knowledge. A chromosome is made up of DNA that is composed of the 4 basic elements, ACGT, abbreviated. And there's genes on a chromosome, and the genes code via a messenger RNA for proteins, and the proteins basically do the work in the cells and in our body. The fact that these genes get transcribed is under regulation. So not every gene on our chromosome is transcribed all the time. Then I think there would be a gross shortage of space in the average cell. But there is very strict regulation as to which genes get transcribed and the proteins expressed at what point in time and under what circumstances. And part of the machinery that regulates this is a subclass of proteins which are called the transcription factors. We usually use TF as an abbreviation there. So there are particular genes encoded in the genome which code for transcription factors, and the transcription factors in turn control other genes. They control the transcription of other genes. That's why they are called transcription factors. So we can think of a chain, a hierarchy of events. And in the cell, I already said, there's a number of proteins in the cell. And nowadays, it is still technologically very difficult to measure the amount of an individual protein in a cell. But what is much more accessible is the amount of a given messenger RNA. That's this intermediate between the gene that's encoded on the chromosome and the protein. And as a result of these measurement techniques, which measure here the messenger RNA rather than the protein, you can think of the cell as a collection of mRNA molecules. And here, the mRNA molecules of the same color are intended to signify that they come from the same gene. And they are expressed in different strengths or levels. So some of them are lowly expressed, and others will be very abundant in the cell. And it's now about 20 years that we have technology to measure these abundances. That started in the 90s with the microarrays. Nowadays, we have a technique that's called RNA-seq. But for the purpose of this, this talk now, it doesn't matter where the data comes from. In the end, we have our mathematical abstraction. We have a matrix with expression levels of the individual genes, where G1 to whatever is our genes. Yeast has 6,000 genes. We have about 20,000 genes. So that's the order of magnitude that we're talking about when it comes to genes. And we have measured the contents of the cell under different conditions or different cell types. You can think of different cells in your body like muscle cells, brain cells, liver, skin, whatever. And each of those cell types would be one row here. It's often, we oftentimes speak of the samples that we have available. And the distribution of the genes will be different in each of these samples. So we are inching our way towards the abstract formulation of a problem. Let's say these are 2 genes here. Each of these columns corresponds to 1 gene, and we have many samples. And red and the one is high and low. So the eye can immediately pick out that there's some kind of anti-correlation between those genes just from the color coding here. And that really reflects the intuition that we have that that since the transcription factors can activate or also repress other genes, there's associations between the genes that we can tell from the abundances. So the columns are those abundances, and we want to tell what the associations are. So here it's called the observation vectors. And really what we want to determine is whether between G1 and G2 there is some kind of relationship or there's no relationship between them. So in an abstract sense, we think of these nodes here as representing random variables, and these are samples from these random variables which correspond to the two genes. Of course, we think that in the cell, in the reality, there are more or less complicated relationships. Yeah? One transcription factor will turn on a number of genes, one of which is again a transcription factor. This gene may be under the control of this one as well as of this one, and this one together with this other one will have an effect on yet another one, and so on. So this is the type of network interactions which we would like to recover only from this data matrix that has been measured. So I said that about 20 years ago, the technology was developed to perform these measurements. And here's a quote from a paper in 2000. Maybe you have made the same experience with your students, that before 2000, there was no scientific literature. So this is from year 0 of scientific literature. And that was became pretty well known, this paper in our field, discovering functional relationships. That's what I've been talking about. Here it's not within the set of genes, but it's between RNA expression and chemotherapeutic susceptibility using relevance networks. Relevance networks was also a term that was, I think, coined in this paper. And it is worth reading. This comprehensive pairwise correlations were calculated between gene expression and measures of agent susceptibility. Associations weaker than a threshold were removed, leaving networks of highly correlated genes and agents called relevance networks. Sounds very straightforward. We do all the correlation coefficients among all these vectors that we saw before, and whenever this is not 0 or larger than some threshold, we draw an edge, and that's the network. And this is a figure from this paper. This is what the result looks like. You can see they obviously do networks for a fairly large number of genes, and they have 60 cancer cell lines. So you have 60 measurements per gene, and this gives rise to this network after selecting a particular threshold. And when you look at this sort of in a more systematic way, here we have again this matrix. The columns are these measurements, all the measurements for individual genes. And in this example, say, the blue and this funny greenish color or so have a non-zero interaction, and that is represented by an edge. And in particular, there's no edge when we deem the correlation coefficient to be 0, or of course, in reality, it's never 0, but it's just very small. So we always have to select some threshold below which we do not draw an edge here. So what's the problem with this? Well, in the world of genes, this is fairly easy to say. Suppose we have one gene that activates another gene, and that gene in turn activates a third one. We will see this in the data, hopefully. But the data will also make it appear to us as if there were a relationship between gene 1 and 3. Yeah, because we have a good correlation between the first two. So here's a quote from another paper. We are now 11 years later. It's not really about gene networks. It's about residue interactions, but the mathematics here is the same. First, the authors say that the covariance or correlation-based analysis, so the networks that I was just telling you about, has a problem because of what it says here, residue belonging to partner proteins of this pathway. It was found to partially predict correct interprotein residue contact, but also many residue pairs which are far apart. A major shortcoming of covariance analysis is that correlations between substitution patterns of interacting residues induce secondary correlations between non-interacting residues. So they have pinpointed the problem, and they are developing methodology to take care of this problem and to eliminate these secondary correlations. So in the statistical literature, these secondary correlations have actually been dealt with not 20 years ago, but about 100 years ago. And what the statistical literature does is they introduce the partial correlation coefficient, exactly for that reason. And what the partial correlation coefficient does is this. Here we have it in the same color code. We first take one variable, say the blue one, and regress the one variable with respect to, say, the green and the brownish one here. Yeah, regress ing on the remaining variables. We also regress, say, the red one, if we want to determine the relationship between blue and red, on the remaining set of variables. And that's where we obtain two sets of residuals. So this is a regression now versus, you can think of it versus a plane of the two variables that are left. And you have all these errors, and you regress these error vectors between the two regressions that were done in here. And a partial correlation is defined. Oh, what a shame. Okay. The partial correlation is defined as the correlation between these vectors of residuals. It's a very straightforward definition. And that, as I said, is about 100 years old. Why would you do that? Yeah? Why would you do that? Let's think about this motivating example again, that I used to explain that there's a problem with these transitive interactions. Suppose I do the regression from 1 to 2 and from 2 to 3. Each time I make an error, and these vectors of errors don't know about each other. They have nothing to do with each other. Yeah? What about the regression from 1 to 3, which happens to be our induced one? When I look at the residual of the regression from 1 to 3 and compare to the residual of the regression from 1 to 2, part of the information is the same, because 2 is halfway towards 3. So these are not independent. These know about each other. Yeah? Likewise, this set of residuals with a set of residuals from here. And this is the insight behind this definition. Yeah? So what does one do? It's the same philosophy. We have these columns here, and now we compute the partial correlations, and that will hopefully tell us that, say, this particular edge here is superfluous because it's induced by the two other ones. And building networks from partial correlations nowadays goes by the name of Gaussian graphical models. And that's part of what one now learns in a class on graphical models. But I promised you that I would also talk about the variance-covariance matrix and its inverse. So how does that come into play? What is the connection with the variance-covariance matrix? This is our data situation here. This matrix here has, say, the genes. Now, actually, I swapped the dimensions. The genes are here, and the columns are now the conditions. And that's the typical situation in biology, where we have thousands of genes, but since for every condition, some poor graduate student has to do an experiment, there's a very small number of experiments and a very small number of columns here. And to get the variance-covariance matrix, we multiply this data matrix with its transpose, and then we get the variance-covariance matrix, at least under suitable normalization that was done beforehand. So our variance-covariance matrix is this product here. The classical correlation coefficient that we know from school can be expressed as an entry in the variance-covariance matrix divided by the geometric mean of these II and JJ entries. Yeah, that's a sort of classical formula. And we do that, and that was implicit in the definition of the relevance network. We do that because when this correlation coefficient is 0, the two variables are stochastically independent. What about the partial correlation coefficient? I said the partial correlation is the correlation of the residual vector upon regressing both x and y to the remaining variables. The remaining variables are here written as this boldface z. I also said this is about 100 years old. It started with a longish discussion between Pearson and Yule that began a little bit before 1900 and stretched on. And it was all under the headline of, and I won't use the word anymore afterwards, it was all under the headline of finding causal relationships. Because when people are looking for causality rather than correlation, they want to know whether a correlation could be due to another cause. And that led to the development of the partial correlation coefficient. Fisher then worked on it and computed the statistical distribution. And sort of the canonical definition is in this early version of Kendall's statistical theory, where Yule was still involved. And what the partial correlation coefficient does mathematically is it uses not the variance-covariance matrix, but it uses the inverse of the variance-covariance matrix. So now let's call the elements of the inverse variance-covariance matrix little p, and then this is the definition for the partial correlation coefficient with respect to the set of remaining variables, so a set of vertices V without I and J. Yeah? And you see it has the same shape, except that the P now is not the element of the variance-covariance matrix itself, but it's an element of the inverse of the variance-covariance matrix. So all this business of correlations and partial correlations can very elegantly be expressed in terms of the variance-covariance matrix. Why do we do it? We had the basic theorem about correlations, that zero correlation amounts to stochastic independence. For the partial correlation, a zero partial correlation coefficient amounts to conditional independence given a set of variables. This is the definition of conditional independence. It's kind of an intricate thing when you think about it. I'll give an example right away. So you see it's very parallel to the classical theorem about correlations. And it also tells us what we are really doing when we do these networks. If we want to avoid the transitive edges, the induced edges, we don't look at the variance-covariance matrix, we don't look at the correlation coefficients, but we look at the inverse of the variance-covariance matrix, and we exclude those edges where the partial correlation coefficient is more or less zero. And that's the theory behind the Gaussian graphical models. So here's an example. This figure was made by one of the students I mentioned in the beginning, and it's really, it really got me started on preparing this set of slides because I like this figure so much, and I wanted to do a talk around this figure. So we have a number of, let's say, genes, any kinds of variables, x1 to x4. Now, suppose they are linked in this kind of chain. And what Juliane, the student, did at the time for her presentation was she prepared a simulation where the first column comes from a standard normal distribution. The second column is a transformation of the first one, where x1 enters into another normal distribution. So the second one is derived from the first one. Likewise, the third is derived from the second one, and the fourth is derived from the third one. That's why it's chain here. So based on this, we get this table of covariances among those four variables. And you can see that all elements are filled. There is no zero element here. So the covariances make it appear as if the network of these four variables would be fully linked, because somehow there's a correlation between everything. When we go on and we invert this matrix, all of a sudden the zeros appear. And the zeros are exactly for those edges which are only induced edges, like x1 to x3 doesn't exist. It's only induced by chaining these generative laws together. And that's why in the inverse of the variance-covariance matrix, this edge disappears and becomes zero. You can also see that you can recover exactly the factors that were put into the laws here. So when you make no edge where there's a zero in the inverse of the variance-covariance matrix, you can precisely recover the chain according to which the data were generated. So here, the independence holds, but in the inverse, what holds is the conditional independence for the 0 entries. And conditional independence now is easier to understand, like 1 and 4 are conditionally independent because if you know x2 and x3, it uncouples them. And then they look independent, but not in the presence of x2 and x3. So bottom line, the recipe to compute networks from this type of data is an extremely simple one. You invert the variance-covariance matrix, with the variance-covariance matrix only being the data by its transpose, nothing else. That's usually called, or often called, the precision matrix or concentration matrix. And the network then has no edge whenever the entry of the precision matrix is 0. And we hope that that way we have no transitive effects. So quick simulation on this. This was the network that we saw in the beginning. One simulates data according to some law. And then if this was the adjacency matrix of this network, and the dark entries is where there is an edge. This is the input data. And when we try to reconstruct it after simulating and adding noise, what we get looks like this. We have this little square up here that is recovered. We have this hook here that we see, but yellow is fairly small number. So we see that things start to depend on where we would set the threshold for what it means to be 0. But in principle, the hook here is visible, likewise here, and this checkerboard pattern down here is also visible in the reconstruction. So the method works, at least on a simulation, amazingly well. Here's an example. I was just trying to give you the impression that people do use this on biological data, although once you start to really work on biological data, there's always various odds and ends that one has to take into account. This figure is taken from the work of these guys, Castello and Roverato, who have a program, a number of papers that follow the philosophy, but are much more refined than what I've been showing here. However, there's a number of problems that one has to be aware of, especially for the applications in biology. I already pointed out to you that in biology, people frequently want to compute networks for many, many genes, although based on a fairly small amount of data, because the data have to be generated. So here's the result of simulations where we generated a network that has 50 nodes. The data are strictly Gaussian. I didn't even add noise to it here. So it's really just to confirm the theory. And the ROC curves are to be understood such that since we generated the data, we have a gold standard network. And when we reconstruct, we can sort by the absolute value of the partial correlation coefficient. And then we can do a ROC curve and see how easy is it to get back the correct edges from the gold standard. And you see it depends a lot on the sample size. 50 nodes. If I have fewer samples than nodes, 30 samples, this is the ROC curve. So we're making quite a few errors here. 70 samples is green, 100 samples is red, and 300 samples obviously is enough data to really make it very, very clear to the program what the original network looked like. I have to sort of make one remark on that, which has nothing to do with this particular topic, but unfortunately has a lot to do with genomics and bioinformatics, and that is the slippery slope towards scientific misconduct and fraud. This was one simulation. Whenever I do this with a new simulation and new random data, it looks very different. I actually did it like 10, 20 times until I had an example that I could really use here for didactic purposes. The proper way to do it, of course, would have been to run it 100 times and do this with means and error bars and all that. But as it stands here, it's almost fraudulent. You very rarely get such beautiful pictures. But still, I mean, to drive the point home. There's another catch here that I want to point out. I was basically trying to bang the classical correlation coefficient into the ground. So what happens if we do this with a classical correlation coefficient and not inverting the variance-covariance matrix? Looks even better. Yeah? So what is going on? Why am I telling you all this? What this says is the sorting of the edges is still good. Yeah? But we need to eventually take a decision which edges in the network are real and which are only induced edges. So when we look at the distribution of the classical correlation coefficient and at the distribution of the partial correlation coefficient, in the partial correlation coefficient, we have this set of edges that have a very, very high, almost one correlation coefficient of almost 1. And all this is 0, and there's sort of a little bit of a twilight zone here. Yeah? In the classical correlation coefficient, you can tell that it would be extremely difficult to decide where to have a threshold for which edges to leave out. Yeah? So there's much more of a smear here on the way towards the good correlation coefficients. So this is really the point. The partial correlation coefficients must disappear for these transitively induced edges. And here we get a very clear view of the edges that we don't want, whereas here it's much more difficult to come up with a threshold. I also wanted to communicate to you that this is so simple and so generic that obviously it must be very, very hard to come up with totally new ways to think about network reconstruction. So one can go through the literature, and it's fairly sobering to see how many novel methods eventually return to what I've been telling you. Yeah? So here's one example. And it's a very good example because I think it drives home the points that we just discussed. This was a paper much discussed in recent years. One half of this very intense discussion came from here, I think, from Berkeley. It's the paper which Leo Pachter and Manolis Kellis were arguing about. But what I'm going to talk about has nothing to do with their blog. It's about a different aspect of this. The paper is called Network Deconvolution as a General Method to Distinguish Direct Dependency in Networks. So direct dependencies that has concerned us now, all this half an hour that I've been talking so far, and he puts forward network deconvolution. And the thinking is neat. He says the data that we actually get in biology, they stem from the following process. Certain genes influence each other, and when we collect the data, we also collect the transitively induced consequences of the original interactions. And we are interested only in the original interactions. So in graph theoretical language, he says, this graph is the transitive closure of that graph, and what I would really want to do is, I want to go backward, invert the transitive closure, he calls it deconvolution, and get the original edges which had given rise to the data that I measured. And he makes the following neat observation. He says, our observed data, so the data matrix that we collect, is the product of the original direct interactions, and then, of course, the sort of first order transitive edges you can express as the square of the adjacency matrix of the graph. So the original direct edges plus the square of the original interactions. Then you have a next order transitively induced edges. Those are the cube of the original adjacency matrix. And if you fully close this transitive closure process, you need to add up an infinite sum here. So that's what one learns in school, that if only a number is small enough, then I have this inversion formula for the series, and this is x times x divided by 1 minus x. So for graphs, it would be the matrix G direct times the inverse of identity minus G direct. Yeah? The whole debate then was about the convergence properties. Under what circumstances does it converge, and so on. But this is the thinking. And in order to recover the original data, the direct interactions, the authors of this paper simply solve this little equation, and then they arrive at this formula here. The observed data times the inverse of identity plus the observed data. So a very neat formula. So now let's try to link this to the variance-covariance matrix. The data that we compute, that we measure, I already tried to convince you. We have a measurement matrix, and usually as a similarity measure on the genes, people use the correlation coefficient or the covariance. So in essence, if we would assume that the data is normalized, and in our case now here also standardized to a standard deviation of 1, then we can just express it again, the similarity matrix, as data matrix times transpose of the data matrix, and we have a variance-covariance matrix again. And now, the authors of that paper, they have one little remark. They say, well, self-loops make no sense. Yeah? We want one gene to influence another gene. So what they do in their program is they set the main diagonal of the observed data to 0. If I do this normalization and standardization, what that amounts to is that I take my variance-covariance matrix, and the main diagonal of this will be 1. And when I subtract the identity matrix, that's how they say that they compute their matrix of observed similarity values. Now let's put this here into their formula. This was their formula. G observed is now variance-covariance matrix minus identity. So G observed is sigma minus identity. And when I put sigma minus identity into this parenthesis here, I have plus the identity minus identity. It disappears. The inversion goes here. And then I multiply the inverse variance-covariance matrix onto this. And in the end, I'm left with the identity matrix minus the inverse of the variance-covariance matrix. So if you now forget the identity, their whole procedure amounts to inverting the variance-covariance matrix. And that's what I meant to say. It is very hard to come up with a totally new method, because this variance-covariance business is so fundamental that it just solves the problem. So in reality, I glossed over a whole lot of really hard problems. You may have heard of the P much larger than N problem. This is what has been driving the development of statistics and computational biology for the last 10, 15 years. This is the shape of our data. We have N conditions and P genes. And I already pointed out to you that there's a certain imbalance when I want to infer information on many, many relationships among genes from fairly small amount of data. And mathematically, you can see this apparently in the fact that when I multiply this matrix with its transpose, the rank of this matrix will be way below the size of the matrix. Yeah? The rank will typically be n, if it's not n minus 1 or n minus 2, which it could also be, but it cannot be more than n. Yeah? That's the maximum of rank for this large matrix here. So again, we have kind of an information problem here. Yeah? The matrix only pretends to contain a lot of information, but doesn't in reality, because it doesn't have full rank. It has way less. So what would we do in this situation here? Yeah? How can we remedy? So option 1, the more statistically inclined about you will know about shrinkage methods. Maybe actually the first thing that you say is, oh, I know how to invert singular matrices. Yeah, we can use a Penrose inverse. We can use the singular value decomposition. That's not a problem. Yes, all these things are doable. They do not work particularly well. the set of shrinkage methods works quite well, and there is a group of methods which use the assumption, well-founded assumption, that we don't want so many edges in our network. So what we want is we want a sparse network, meaning we want many zeros in the inverse of the variance-covariance matrix. So they compute the inverse of the variance-covariance matrix in a way such that there will be many zeros there. So those are the sparse inversion methods. That's all very interesting stuff. Actually, since I had the luxury to spend time here, I did a lot of reading on this in the last couple of months. But then there's the other option, work on another problem. Yeah? And this is what we've been doing. We looked at chromatin data. Why look at chromatin data? Chromatin data turns the data situation around. So let me explain how that works. What is chromatin? In eukaryotic genomes, we are eukaryotes, for example. In eukaryotic genomes, the DNA is tightly wound into little knots in our cells. The reason probably being that otherwise the volume that the DNA would take up would exceed the possible volume of a cell. So in order to really compact the huge amount of DNA that we have this very intricate mechanism of compacting it. So the DNA is wound around this set of proteins here in the middle, and this whole thing is called the nucleosome. And a set of proteins here in the middle, they are called histones, several of them. They have part of the chain dangling out of this complex. And there are little chemical moieties attached to these chains. These are the so-called histone modifications. And there's a whole number of different chemical modifications. And accordingly, there are many different histone modifications. People have studied on the order of 30, 40 such histone modifications. Probably there's more out there. And people also observed that the histone modifications in the regions where the genes start, in the promoters, have a relationship to the expression of the genes. They are somehow involved in the regulation. So for example, what do we have here? Histone 3, that's one of these proteins, lysine 4. So interesting, the amino acid lysine is abbreviated with K. Lysine 4 trimethylation is very strong in the promoter region, that's the beginning of the gene, and is positively correlated to the process, to the induction of the gene, to its expression. That's one such example. And in the recent years, people have determined the levels of these histone modifications in the genome for many different cell types. So this K562 is a cell type. H3 lysine 4 monomethylation is one of the histone modifications. And this is the amount of this modification along the DNA. Up here we have a gene. The transcription starts here. So this particular modification is present not only here in the beginning of the gene, but also in what we say the gene body. This other one, H3 lysine 4 trimethylation, I just pointed out to you that this is connected to transcription, and it tends to be high at the beginning of the gene in the promoter. H3K4 trimethylation, we see exactly here in the promoter region, and then it disappears again. The same holds true for this K27 acetylation. And here something is totally absent. This 27 trimethylation is traditionally seen as a hallmark of repression of a gene. If we would have a peak here, the gene would be off and would not be transcribed. But the other situation is the one that we observe here. The activating marks are on and the repressive mark is off. So you can already tell there is relationships also among the different histone modifications. So what we started to do as these data appeared, we started to ask questions about what is the relationship among the different histone modifications. How can we approach this? So this is now an entire genome. It consists of 3 genes. Each gene has sort of the gene body from where the messenger RNA is transcribed, and it has a regulatory region that's the promoter. In this regulatory region, in our case, it was the laboratory of Keiji Zhao from the National Cancer Institute in Bethesda, has determined around 30 different histone modifications. So we basically look at this region here, and we sum up, or we compute the integral under this curve here in the promoter region. That's the meaning of summing the tags in the promoters. So we get one number per histone modification and promoter. And that means a vector of like 30 modifications per promoter here. Now, let's look at the data situation in this problem. We have a small number of histone modifications, and the thousands of genes in an organism, each one has a promoter. So we have a huge number of promoters available. So we have solved our data shortage problem. Yeah, we have enough data now to actually compute the variance-covariance matrix and also compute its inverse. Our P by P matrix now, the P is much smaller than N, and we don't need to fight with the P much larger than N problem anymore. So here is the correlations among the different histone modifications. And you can see, for example, up here, a correlation coefficient of 1 is almost white here. Up here is a cluster of highly correlated histone modifications. And here we see this H3 lysine 27 acetylation, which is a hallmark of the activation of a gene. So all these are correlated with the activity of the gene. Whereas down here, we have the 27 trimethylation, which is the hallmark of repression. So this whole cluster down here is a cluster of histone modifications that are connected to repression of the gene. But among these clusters, we are still interested in what really are the mechanistic interactions, because we think of the mathematically direct interactions as being the one that are biochemically realized in the cell. So this is the matrix of partial correlation coefficients, and it's much sparser. The red is now the good ones, and 0 is around here, this turquoise. So many, many entries here are near 0, and we can compute networks. And then we get a network that one can actually look at and study and compare to the biochemical information. For example, here, blue now means anticorrelation. We have a thick blue line for an anticorrelation between the activating mark and the repressive mark. This is exactly what I pointed out to you based on the tracks of the individual modifications. Also, the math doesn't care about where the data actually come from. I just included the levels of the messenger RNA into the analysis, just as if it were a histone modification, although it isn't, but who cares? Yeah? So the level of the mRNA expression is inversely proportional to the repressive mark and positively correlated to the activating mark, just as it should be. And then sort of one can go through this and interpret it like there's a whole cluster of other acetylations here, but it is interesting to see that those are far away from where the real regulatory activity is. And then we have pushed this farther. There's also proteins that are responsible for putting these chemical moieties, the histone modifications, or for removing them. So there are enzymes that actually do the processes. And again, we have data for their presence in the promoters, and we are interested in which of these interactions are really direct, because that's where the biochemistry is. So we did the same analysis with the partial correlation coefficients to combine enzymes, like here this PHF8 or SUS12, to modifications, which are here the little rectangles. So the more biologically inclined will recognize this, for example, as the famous polycomb complex, that is made up of a complex of EZH2, SUS12, and one or two of these CBX proteins. And this complex sets the H3 lysine 27 trimethylation repressive mark. So one sees this immediately from the data here. And the other thing that one does is one can compare, of course, to the literature, and all the solid lines here are known, have been reported in the literature as being real interactions. The dashed lines here are not known. So a collaborator, which was Ho-Ryun Chung, also in our institute, set out to verify these experimentally, because the method was really suggesting that there should be an interaction between this H4 lysine 20 monomethylation and these two proteins here, which was not known and is actually, to the biologists, fairly surprising. And when one does the experiments, one can really verify this. So there's new knowledge that is coming out of this type of data analysis. Nevertheless, I do want to point out that the machinery that I was summarizing for you is not limited to these biological applications. I did a little bit of Google Scholar searching, and here we have on the inverse of the covariance matrix in portfolio analysis in the Journal of Finance. Or we have partial correlation for functional brain something in functional MRI, in neuroimage, biological cybernetics, bioinformatics, as I was talking about. So it's all over the place, and essentially always the same machinery. It's very nicely described, all of this, in the book by Hasty, Tübscherran, and Friedman, Elements of Statistical Learning. But even there, it only made it into the book in a second edition. Yeah? The first edition did not yet contain that. The second edition has a chapter 17, I think, which summarizes what I was just talking about. Two people from my institute a couple years back wrote a very nice review where some of this can be read, and this is the pointer to our work where we have used it on a data situation that is more amenable to this type of analysis. And with this, I'm done, and I thank you for your attention. Yes.
So, if you actually look at the joint distributions of genome activity levels and so on over all these trials, how well is that joint distribution modeled by multivariate Gaussian? I mean, the Gaussian assumption is very nice and practical. How accurate is it in practice?
It's totally violated. What we've done in our own work is, a few years back, we used rank transformation. So we rank transformed the data, which means that you compute a rank correlation coefficient and a rank partial correlation coefficient. And that partly takes care of it. And actually, this is also one of our current research activities, how we can generalize all this to become independent of the Gaussian assumption. So my hope is that within a week there will be a paper on arXiv with our answer to this. But it has bothered us a lot, and we spent quite some energy on this. But I didn't want to make it a topic for this presentation here. Oh.
So you deliberately avoided the term causality. Can you comment?
Yeah. It does. I mean, when we look at the data, the partial correlations tell us, I think, indeed a lot about the causality. You have to assume that you have more or less complete data. Yeah, that really the physically interacting variables are present in your data, and if that is the case, then you can ask, if I have a correlation that gets explained away by another variable, usually that really has to do with biochemistry that is going on there. Yeah? We are now currently experimenting with Bayesian networks on the one hand, but there's also a very interesting paper by Peter Bühlmann where he combines Bayesian networks with Judea-Pearl's do calculus to determine the direction on the arrows. And what we are again currently doing back in Berlin is we are using the histone modification data to test all this machinery, because for the histone modification data we have this classification into readers, writers, and erasers. So we have biochemical knowledge about the direction of the error of the causality. And so we are using now the IDA method, it's called, from Peter Bühlmann, on this data and trying to understand in how far it is possible to recover what we know biologically. The short answer is about 2/3 of the directions we get right. Is that statistically significant? Hard to say. We only have this one example. We'll see. We are still working on that. Yeah.
So if you have already a network, like in the example of Chromatin, so we have already one existing network, and then if we compare the correlations that we get, the Gaussian correlation, and already the edges that are in the network, are they concordant? So we always have, if we have positive correlation, we have edge.
They are not concordant. There's a whole literature on this. That you can construct terribly contrived cases where the partial correlations have the opposite sign of the correlations. It gets very confusing. You cannot rely on the sign of these to give you sort of the intuitive answer. There's a book that's called Path Analysis and something. I can look it up for you, which goes through all these contrived examples and warns you. One of the reasons actually to move over to the inverse of the variance-covariance matrix and to these shrinkage methods to estimate this is that the partial correlation coefficient in itself is numerically very unstable. Because if you have vectors where two of them are highly correlated, then you start in the definition of the partial correlation, you are near a singularity. So it's numerically extremely difficult for a large set of vectors to compute this. And that's much of the reason why we need these shrinkage methods. Otherwise, the naive computation just goes off into space somewhere. Yeah.
So for the histone notification data, the measures are along the genome, right?
Yes.
So the popular approach has been to train, let's say, HMM or a model along the genome to try to model the dependencies between successive positions. What you do is different, right? You fix one position and you forget about the othering and you find the link between the variables. So it makes sense to combine both and then make a network to include the dependencies of previous and next steps.
It's an interesting thought. Yeah, I need to think about this. Maybe we'll talk about it one of these days. It's an interesting idea. I hadn't linked it in my head, to be honest. Yeah.
Related to that, do you think there are combinations of modifications in different places in the genome that may have different behavior?
I believe so, yes.
So I guess your network, do you use kind of the average or is it global across all the data?
Well, we kind of work under the assumption that promoters tend to be similar to each other, but there are other parts of the genome. We have another paper that appeared recently in Cell Reports where we did the segmentation and we did networks for each type of region. So there's a network for enhancers and there's a network for promoters. And then, of course, you see somewhat different networks, which I think, again, reflect the biological function of these individual regions. I never dared to go into heterogeneity within one type of region.